Today I'll be speaking about ACD's multi-omic spatial technologies and their applications in RNAi therapy. In this webinar, I will start with an overview of the RNAscope in situ hybridization technologies that we have developed at ACD. Then we will take a closer look at our assay portfolio, followed by detailed introduction and applications of microRNAscope and RNAscope Plus assays. And finally, I'll provide a brief information regarding ACD's CRO division, which is called as Professional Assay Services or PAS. More than a decade ago, in 2012, we first published the RNAscope technology and demonstrated the co-detection of multiple transcripts with single-cell resolution by this novel in situ RNA analysis platform. Since then, RNAscope technology has been mentioned in more than 10,000 peer-reviewed, high-impact papers, and the number is still growing. We have been established as the experts in the field of spatial transcriptomics, typically with single-cell resolution. At ACD, we have about 300 team members with more than 60 PhD scientists who are equipped and well-trained to interact with our customers, understand their scientific need, and provide support in real time. Our quality management system follows ISO 13485 standard that is required for selling medical devices. All probes and reagents are manufactured under the strict ISO 13485 guidelines for use in clinical applications. RNAscope has been also used as CDX tools for several clinical studies in the past. The original RNAscope technology was developed to detect endogenous mRNA transcripts. However, with time, we understood the need for spatial visualization of many different nucleic acid molecules. With continuous research and development work at ACD, now we are enabling our scientists to detect all sorts of targets, which include endogenous and exogenously delivered molecules. This is possible due to our unique, specific, and yet flexible probe design strategies. We say that probes are the heart of our technology, and by using right probes, we can visualize mRNA, long non-coding RNA, short sequences such as siRNA, ASOs, or microRNAs, and RNAscope can even detect vector or any plasmid or episomal DNA. However, the events at the level of genomic DNA, they can be detected by using DNAscope assays. So far in our catalog, we have more than 50,000 probes which are ready to ship and use. In case you don't find a probe in our catalog, our dedicated probe design team will design one for your specific target. And for any proprietary sequences, a probe can even be made exclusive for the requester, meaning that it will not be added to our catalog and all information will be kept confidential. We can say that RNAscope assay offers visual analysis of any gene expression with morphological context, especially with lower to mid-plexing capabilities. The image shown here is obtained by 2-plex chromogenic RNAscope assay on non-small cell lung carcinoma tissue. The green dots, they represent PD-L1 expression, and the red dots are showing expression of PD-1. Clearly, just by looking at the image, one can identify the heterogeneous expression of these targets in this tissue sample. Different regions in the same tumor tissue section is displaying different levels of PD-L1 that you can visualize as green dots, and they're ranging from low to medium and high expression, pointing to an intra-tumor heterogeneous expression pattern for PD-L1. Similarly, the expression of PD-1, which is shown as red dot, is showing that it is mostly confined in the stromal region, and these details are actually easily lost in any bind and grind assays, or it gets difficult to interpret in ultraplex spatial assay. Since the RNAscope data is quantifiable, it provides a clear quantitative measurements on ROI-dependent expression levels. As shown here, RNAscope assay followed by software-based quantitative analysis is used to tease out varying gene expressions of PD-L1 and LAG3 in tumor microenvironment. ROI-specific analysis showed that in this tumor, PD-L1, which is shown as green dots, is primarily expressed in the tumor compartment, whereas LAG3, which is shown as red dots, is primarily expressed in the stromal compartment. And this expression pattern can be visualized in the stained tissue section, and it can also be quantified by halo analysis. The right bar graph is showing quantitative data obtained with halo analysis of PDL1 and LAG3 gene expression in tumor and stroma of the non-small cell lung carcinoma tissue specimen. As noted before, such detailed ROI-specific information is often lost in the bind and grind methods such as qPCR. Our unique probe design and signal amplification system allows RNAscope assay to be highly sensitive and specific. Let's dive a little deeper to understand the technology. The oligonucleotide probes can be conceptualized as these to emphasize the fact that they have two regions linked by a spacer. The bottom of the Z, it complements and hybridizes to the target molecule, and the top of the Z portion, it is utilized for the signal amplification. These two parts are linked by a middle portion that is called linker or spacer. When two Z's, they hybridize in tandem to the target sequence, it creates a unique binding site for a preamplifier. On each preamplifier, multiple amplifiers are allowed to bind, which in turn are labeled by chromogen or fluorophores. This signal amplification strategy yields high sensitivity and allows for visualization of target RNAs as a single dot. The step-by-step addition of smaller components allows easy entry into the cells, and much larger final amplification tree, it ensures sensitive and persistent detection of target. Non-specific, unbound smaller components are washed out effortlessly. A standard RNAscope probe consists of a pool of 20 ZZ pairs, and each one of these ZZ pairs, they can generate signals. By incorporating the large number of these ZZ probes, it allows for a tremendous signal amplification by high level of redundancy. Although binding of 3 to 4 ZZ pairs can generate detectable signal, the sensitivity of the assay, it comes from the redundant signal amplification by all of these 20 ZZ pairs. While the assay is highly sensitive, it is extremely specific at the same time. The specificity of the RNAscope assay, it comes from the fact that binding of a single Z, it cannot support formation of stable amplification tree. And just in case one Z binds non-specifically to any target, this amplification tree will not be built and therefore signal will not be detected. This is a slide showing various assays applicable for detection of transcripts, short sequences, and episomal and vector DNA. The selection of one assay over the other is mainly dictated by the size of the target sequence. RNAscope is the assay best suited for targets with more than 300 nucleotides. It offers maximal flexibility with options of chromogenic or fluorescent readouts. RNAscope assays can be performed at singleplex, multiplex, and high-plex capabilities. BaseScope is the assay which is ideal for targets between 50 to 300 nucleotides, and it is frequently used to distinguish highly homologous sequences and identification of point mutations as well as splicing events. It can have single or duplex readout with chromogenic labeling. MicroRNAscope assay is a choice for short sequences with 17 to 50 nucleotides in length. These are mainly applicable for siRNAs, ASOs, microRNAs, or any other short oligonucleotide sequences. RNAscope Plus, on the other hand, is utilized to detect one short sequence in combination with up to 3 mRNAs. Please note that our assay workflows are compatible with protein code detection, and the protease-free workflow makes it a seamless process to visualize protein and RNA on the same sample. Let's talk about the assay workflow. This is a slide-based assay which can be performed on a wide variety of sample types, including FFPE tissues, fresh frozen or fixed frozen tissues, as well as cultured cells. And the first step is to permeabilize the cells or tissues, followed by probe hybridization and signal amplification. The signal is generated in the form of punctate, chromogenic, or fluorescent dots, and it can be quantified. Please note that the microRNAscope probes are not based on ZZ design. However, the signal amplification method is the same, allowing the best sensitivity to the assay. Images on this slide are the typical chromogenic and fluorescent data obtained with RNAscope, and each individual dot represents one target molecule, and therefore counting the dots provides the number of target molecules per cell. It is important to remember that the number of dots are quantified, not the size or intensity of the dots. Since the focus of today's webinar is the applications of ACD's technologies in RNAi therapy, next few slides are going to be concentrated on microRNAscope and RNAscope Plus assays. As mentioned before, these assays are mainly used to visualize safe and efficient delivery of therapeutic siRNAs or ASOs to target cells. They are also used to measure the potency, stability, efficacy, and toxicity of the therapy. The microRNAscope assay is a chromogenic singleplex assay, which can be performed manually on a benchtop or automated by using an autostainer. These assays are frequently used to validate the delivery and biodistribution of siRNAs or ASOs. MicroRNAscope assay is also used to detect microRNAs and to study their expression and subcellular localization. The de-identified mouse liver data on this slide show the specificity of microRNAscope assay. The mice were treated with either PBS as a negative control or one of the two different siRNAs, number 1 and number 2. The liver tissue sections obtained from these mice were used for visualization of delivery of the siRNA by using microRNAscope probe targeting siRNA number 1. As you can see that the signal is only observed in sample treated with siRNA number 1 and not in the PBS treated or the siRNA number 2 treated samples, indicating successful delivery of the siRNA and also the specificity of the microRNAscope assay. This is the data from RNA journal published in 2023, where the authors demonstrated a specific detection of ASO in murine, kidney, and liver tissues, which were treated with ASO and not in the vehicle-treated tissues. By using a spatial method of detection, the authors were able to delineate the biodistribution pattern with great details. For example, in kidney tissue, the ASO distribution is mainly in the proximal renal tubular epithelium and parietal cells of Bowman's capsule. In liver tissues, the ASO signal is present predominantly in the non-parenchymal cells, including Kupffer cells and liver sinusoidal endothelium, and to a lesser extent in the hepatocytes. Clearly, this level of detailed biodistribution analysis with minimum possible error is not possible with any bind and grind methods such as qPCR. Since our assays are compatible with RNA protein co-detection, the authors performed IHC to visualize ASOs as red signal by microRNAscope assay and outline the hepatocytes as green signals by IHC using antibody for sodium-potassium ATPase in the mouse liver tissue. The IHC data showed high level of cytoplasmic ASOs as seen as red signal in the non-parenchymal compartment. The message I'm trying to convey here is that by using cell markers, either as RNA or as proteins, we can visualize cell-specific delivery of the RNAi therapy. As we can imagine, when an siRNA or ASO is delivered, the researcher would like to confirm the delivery of the therapy as well as the effect on its target mRNA. And one way to look at the siRNA and the target mRNA is to perform microRNAscope to visualize siRNA and RNAscope assay to detect mRNA on serial sections. This slide shows the data obtained with this type of approach, that is microRNAscope and RNAscope on serial sections from the same tissue. The image shows that the vehicle-treated top panel has no siRNA signal and high expression of the target mRNA. The bottom siRNA-treated panel is showing presence of the siRNA and the corresponding reduction of the target mRNA expression. The microRNAscope assays are very useful to study the dose-dependent delivery and efficacy of ASOs. This is the data which was presented by Stoke Therapeutics at ASGCT in 2022. They delivered the therapeutic ASO to modulate splicing event in order to precisely upregulate functional OPA1 mRNA and therefore OPA1 protein in cynomolgus retinal ganglionic cells. On the left side, microRNA scope data is showing a dose-related increase in the therapeutic ASO drug in retinal ganglionic cells. On the right side, the immunofluorescence assay is being used to confirm the dose-dependent increase in OPA1 protein. I would like to take this opportunity to remind everyone here again that it is possible to detect ASO, mRNA, and protein on the same sample on same slide to assess the co-localization. Coming on to RNAscope Plus assay, which is a multiplex fluorescent assay, and it can be used to detect one small RNA such as microRNA, siRNA, or ASO in combination with up to 3 mRNAs. The image shown here is a representative data obtained by targeting an endogenous microRNA, that is RNU6, along with 2 mRNAs, that is PPIB and POLR2A. The white signal represents RNU6 and the red dots are showing the expression of PPIB mRNA, whereas the green dots represent POLR2A mRNA expression in these cells. This is a de-identified data obtained by RNAscope Plus assay on siRNA-treated mouse liver tissue. The assay was intended to show delivery of the siRNA in a specific cell type and also to show the effect on the target mRNA. So 3 probes were used, one for siRNA, the second one to target the cell marker, and the third one to specifically bind the target mRNA for which siRNA was delivered. And it can be clearly seen here that the top PBS treated panel has a lot of mRNA signal, which is shown as red dots. And no siRNA was visible in this PBS treated control sample. But siRNA, as can be seen as a white dot, they are visible in the bottom siRNA treated panel. Also, in the siRNA treated sample, there is much reduced red signal. That means that the delivered siRNA successfully downregulated its target mRNA. And the green cell marker signal is useful in identifying the siRNA delivery and its efficacy in specific cell type. This is another example in brain tissue where microRNA 124 expression is being detected with 3 mRNAs, POLR2A, which is shown here as green, PPIB, which is shown as red dots, and UBC, which is shown as pink dots. And the benefit of fluorescent assay is that you can look at all markers at once to look for co-localization, or you can shut down channels and just look at 1 or 2 of them and look at the co-localization or individual expression in the sample. Please note that microRNA 124 is expressed mainly in brain and not in colon. The RNAscope Plus data on this slide in colon tissue, it is not showing any signal for microRNA 124, which confirms the specificity of fluorescent RNAscope Plus assay similar to chromogenic microRNA scope assay as shown before. This is a similar example showing expression of microRNA 205 and associated tumor target genes in head and neck cancer tissue by 4-plex RNAscope Plus assay. The assay was performed by using 1 microRNA probe to target microRNA 205 and 3 mRNA probes targeting pan-CK, P10 and TP53 in tumor, and nucleic acid were counterstained with DAPI.